as well if I hear some strange noises in the background. That's also very, very good here. So then let's jump right in. Let me introduce Donna Werling, obviously from Stefan Sanders' group here, UCSF. She will give us an overview about her work with the title that everybody has probably read already. She's working PGC autism, I assume so, and would be great if you give us a couple sentences about yourself as well as an introduction. And then also what I also wanted to mention is that you please give us maybe after 20 minutes and after 40 minutes, a short time for questions. Usually the people are not super brave to ask many questions here, but maybe there is somebody who will jump up here. An analytical framework for whole genome sequencing, association studies, and its implications for autism spectrum disorder. So Donna, it's yours.
Great. Well, thank you so much to Stefan and Naomi for inviting me to present today. I'm very happy to share this work with all of you. Like Stefan mentioned, I am a postdoc working with Stefan Sanders and Matt State right now at UCSF, working on gene discovery in autism and functional genomics of autism, trying to understand the biology of autism from a genetic perspective. And that may be all that's relevant to know about me at the moment. So let's dive in. Let's see if I can share my screen. Let's go with this one. Can we all see that, hopefully?
This looks good. Full screen, even better.
Okay.
Perfect.
Great. All right, so before I go any further, I just would like to emphasize, to make the point that the work that I'm presenting is the result of a collaboration of a large number of individuals and groups across the US. And we're all affiliated with the Autism Sequencing Consortium. So I'm speaking on behalf of all of these groups. These are the PIs from this work. So this is not my own work by any means. It is shared. So just a brief introduction. So we're studying genetics of autism spectrum disorders. And as we've seen in the past from exome sequencing in autism, this work was aimed at gene discovery, just try to find genes that are involved in autism risk. And as we've seen from exome, there is an excess of de novo loss of function mutations in autism cases as compared to their sibling controls. So you can see that in the bar plot on the left. These are more frequent in cases than controls. And when we start to see de novo loss of function mutations stacking up in the same gene from unrelated individuals, then we're able to statistically associate specific genes with autism risk. And that's how exome sequencing has led to gene discovery in the past. Now, the gene discovery work that has come out of exome sequencing has now identified a fairly large number of autism risk genes. The most recently published set of high confidence risk genes included 65 genes, although this list is growing as we speak. And those 65 genes are shown in this network plot on the left. And the power of gene discovery for disease is that it can tell us things about the biology of autism or a given disease that we may not have suspected going in. So what this figure illustrates is that there are two general clusters or networks of these risk genes that associate with certain functions. So there's a cluster of genes involved in synaptic function, and the involvement of the synapse in autism risk had been hypothesized for a long time. But then there's this other module or network of genes that have to do with chromatin remodeling, chromatin state, gene regulation. And this involvement of chromatin biology in autism was not known until we had done gene discovery in autism. So this is the power of gene discovery to teach us about the neurobiology of these conditions. Now, this is a pretty high level observation. We have a set of genes. We've been able to sort it into two pathways, but there's a great deal that we still would like to understand about autism biology, namely when, where, what cell type and development are these genes expressed and having their key functions? How do these two networks of genes relate to each other? How does mutations in chromatin genes affect the synapse and vice versa? So there's work to be done going forward. Now, with regard to that understanding of spatiotemporal expression patterns of these genes, when these risk genes are having their key impact, we need to start to turn to non-coding elements. So non-coding regulatory elements. Enhancers, and to a lesser extent promoters, are very specific to cell types and specific times in development. And this is a classic figure shown here from the Vissel et al. Paper in 2009, demonstrating the expression of the same reporter gene under different enhancers. And you can see that each non-coding enhancer leads to a very distinct expression pattern of these genes. And we hope that as we move into the non-coding space for autism, we're able to discover mutations in these non-coding elements that will lead us to a greater understanding of the spatiotemporal specificity of risk for autism, as well as down the line, perhaps far down the line, allow us to deliver very specific, very targeted therapeutics. That would be the hope. So the data and the analysis that I'll be sharing with you today comes from a recently published paper from our group. I think it was published last month in Nature Genetics. So I don't cite this study on every slide going forward, but this is the paper that it comes from. And I'd just like to give credit to my 4 other co-first authors now before I go any further: Harrison Brand, Junyoung An, Matthew Stone, and Lingxu Zhu. Now, right, so as we're moving now from exome to whole genome sequencing, which of course gives us the opportunity to identify non-coding mutations directly. And the study that we did here was based on 519 families from the Simon Simplex collection for autism. These are all quartet families, meaning that we have 1 affected individual, 1 unaffected sibling, and 2 parents from every family. And the Simon Simplex collection is larger than these 519 families, and then this first cut of families was selected from that larger set for the absence of known de novo loss of function mutations in the proband or large CNVs in the proband, with the intention that this selection process would enrich our sample for non-coding risk variants. So the sequencing for these samples were done at the New York Genome Center. We then identified SNVs, indels in this data set, and our very close collaborators, Mike Talkowski's group, handled the structural variation piece, which was a tour de force on itself. But I will be focusing this presentation on the SNVs and indels going forward. So we identified variants, we ran a series of filters to identify a high quality set of variants. From this set, we called de novo variants from this set and wound up with the numbers that you see at the bottom, which are about 64 SNVs per child and about 6 indels per child. And this is across all autosomes with very high validation rates. So we're quite pleased with this the high quality filtering procedure and our calling procedure for these variants. So now we have variants called, but the question is how to interpret them. This is the key question. So in coding regions, we have a very clear hypothesis of which variants mediate risk. It's because of the triplet code, it's very easy to identify which variants will be loss of function, which will be missense, which will be silent. It gives us a very easy way to separate the signal from the noise. In non-coding regions, that becomes less clear. Do we try to focus our analyses on introns, on UTRs, promoters, enhancers, in whatever way you might decide to define those relative to genes? Does it make sense to start adding in markers of non-coding function, for example, epigenome markers, histone marks, transcription factor binding sites, open active chromatin? Do we need to look at highly conserved regions versus non-conserved, human accelerated regions? Do we need to focus our analyses on regions in and around genes that have specific functions, gene sets of interest to us? And the more we started to consider this wide range of possibilities, the the number of plausible hypotheses we could come up with of regions of the genome to focus on just grew and grew. There are any number of very reasonable sounding hypotheses one can make from beginning to combine these layers of annotation. And so that led us eventually to this approach that we call a category-wide association study. And here, I'll come back to the slide again in a few slides on, but the focus here is that we took our variants de novo variants that we called, and annotated them against these 5 different categories or classes of annotation. So can you see my mouse if I show that? Are you trying to? I don't know.
Yes, we can see the mouse.
Great, thank you. So we looked at conservation. Are these variants conserved above a certain FAST-CONS or phyllo-P threshold? We grouped the variants by their type. Are they SNVs, indels? We looked at, we'll call this gen code annotations or gene-based annotations. Where, relative to annotated genes, do these variants fall? Are they coding? Are they non-coding? Are they UTRs, etc.? We also looked at functional annotations, like I mentioned, human accelerated regions or ENCODE data, DNase, transcription factor binding sites, annotated enhancers, ROADMAP epigenome data for chromatin state. And finally, we looked at a number of gene lists. So if a variant, if the nearest gene to a variant was in one of these lists, autism spectrum disorder risk genes, FMRP target genes, etc., then we would consider that annotation. And we combined each of these classes of annotation in every possible combination. So each variant could be annotated to as many as 66,402 combinations, about 52,000 of which are non-redundant with each other. And then the idea was within each of these annotation categories or combinations of annotations, we would run a burden test to see if there were more variants in this category in cases than controls. And the idea here was to run this analogously to a GWAS. So instead of coming in with a candidate annotation, much as one would come in with a candidate gene, we decided to try to test the genomic space, including non-coding regions, as completely as possible, and let the burden of variants lead us to the category of annotation that matters the most for autism risk. Now, this is probably running these 52,000 annotation categories is really the upper limit on the number of things that we can test, and we did run a series of tests at more limited selections of annotation, and I'm going to go through each of those now, and we'll come back to the category-wide association study in a few slides. So what I'm showing here is an overview of our analysis framework that we used on the whole genome sequencing data, maybe the COS, which is the most predominant analysis here, but we've run a series of things here. The tests across the top are those that are category based, looking for a single category that shows a significant burden in cases versus controls. And the tests on the bottom are those that aggregate signal across categories in a more polygenic risk score-like way. But, so we'll focus on the very first test. We do one test looking for non-coding burden. And to run this test, one of the very first things that we observed is that correcting the rate of de novo mutations per individual for paternal age is critical to be able to observe a locus-specific burden in cases that you can attribute to the biology or risk of autism as opposed to just paternal age. So this plot here is showing the very close, very highly correlated relationship between the father's age at birth and the number of de novo variants identified in each sample. Now, in our cut of the Simon Simplex collection in these 519 families, 56% of the cases have older fathers than their unaffected siblings. And this slight skew in older paternal age among the cases, we were afraid might lead to the overall observation of a larger number of of de novo mutations in cases versus controls in ways that wouldn't be meaningful for risk. And so what we did is illustrated by this bar plot here, is to simply adjust for paternal age using linear regression. And so you can see in this plot on the left that we are observing in the raw variance that we called a greater number of variants in cases versus controls, and this is significant by a binomial test. But after adjustment, this difference is no longer significant. And if we restrict that analysis to non-coding only, we see a very similar phenomenon, perhaps because the vast majority of variants that we discover from this are non-coding. So that's a critical first step, and also gets at this first test. When we run a simple test looking at the burden of de novo variants in the non-coding region, we see no difference between cases and controls. All right. Next, we looked at gene-defined regions as a limited set of categories of interest. So here, what we've done is essentially pull out key categories from what we've called the GenCode annotation, and you can see them listed in this plot here. We didn't compare loss of function variants because of the way this sample was selected to have no loss of function variants in the cases, or at least not known. But we do look at missense, damaging missense, silent, promoter, UTRs, splice site, intronic variants, link RNAs, antisense, and intergenic variants. So everything from promoter down are non-coding. And the x-axis on this plot is showing the relative risk, or the ratio of the number of variants observed in cases versus controls. So if you are moving toward the right, that's what we're looking for. We're looking for categories that are enriched in cases. And the red lines in each plot are showing you what we observed in our data, again, after correction for paternal age. The violin plots in each case are showing the results from 10,000 case control label swapping permutations as a way to generate a null distribution, what you might expect to see by chance. And from here, you can see only one of these categories is starred, indicating a significant shift in one direction, or the other. Intergenic variants are showing a slight bias toward cases, but when we correct for the 11 tests shown here, this difference is no longer significant. We then thought to check these same regions of annotation near or in autism risk genes. So for this, we used a set of 179 risk genes associated with autism at a false discovery rate of 0.3. So if the nearest gene to a variant was one of these genes, 179 genes, then it was included in the analysis. And here, what we're seeing is that only missense and damaging missense variants show a case significant burden in cases. And when we correct for the 8 tests shown here, only the damaging missense variants are significant, which in a study that was designed to look for non-coding variants is perhaps a bit disappointing. Yeah. But I mean, I guess it is reassuring that we see a similar signal here that we would see in exome. But again, it's not the frontier that we intended to go for. Before I go on, are there any questions? We're about 20 minutes in.
If somebody has a question, please unmute yourself first. Maybe I have one short question about the age of the father. I mean, here, with the setup of these quadruples, you always have the control to the case, right? Yeah. So does that mean that most case siblings are younger than their controls?
Most case siblings are older than their controls.
Older, that's what I mean.
Yeah.
Exactly. So yeah. That's exactly the case here, and that's why you have to control for that, yeah? Yeah. Okay, makes sense.
That was our thought.
Yeah, perfect. Somebody else here. So, and maybe like if you go back to this category slide, I think the one couple of slides back, the one thing like you say on the left side, test multiple hypotheses of non-coding annotation, and in the end you test coding annotation, right? So
we include coding, that's true.
Yeah, exactly. This might be a little bit confusing, but that's fine.
Yeah, very astute observation.
So you are much less volume right now. Did you change anything, or is this on my side?
No, I did not.
Maybe you come closer again, then it's fine.
Okay. Is this better? Or the same?
So nobody else here? Then let's continue, please.
Okay. Great. So again, I have this overview slide. We're going to skip over the 200 test independent clustering bit and come back to that. We'll move ahead to the category-wide association study. Okay. So run over these annotations. And like I said, every annotation considered in combination gives us up to 66,000 combinations, 52,000 of which are non-redundant with each other. An example of one annotation is shown here in the corner, which includes a combination of these annotations. So de novo variants in DNA's hypersensitive sites, of introns, of FMRP target genes. You can imagine swapping out any one of those annotations and getting a different set of annotations making up a new category. Now, of these 52,000 annotation combinations, 13,700 of them had at least 7 variants in them, which we determined was the minimum number of variants required to have a chance at observing a significant result by a binomial test. And so these are the categories that were taken forward in this analysis. So we're testing about 14,000 combinations. Now the results from a COS analysis look like this. It's a volcano plot where each dot in the plot is an annotation category. The y-axis is the minus log 10 of the p-value for that category. And then the x-axis, much like the violin plots from before, is the relative risk, or the ratio of the number of variants observed in that category in cases versus controls. And so what we would hope or expect to observe in this case is our categories that are enriched in cases. So on this right-hand side, shooting up toward the most significant observation possible. But to be responsible scientists, we need to apply a multiple testing correction for the vast universe of tests that we've run. So what we really hope to observe are categories that pass this multiple testing correction. So for those who have read the paper, this is no surprise that when we look at our results, there is no single category that reaches significance after our correction for multiple testing. And this is the data as it looks. So up here is the multiple testing corrected significance threshold, and a full order of magnitude below that are our most significant annotation categories. Way down here is an uncorrected significance threshold of 0.05. And I think that there are a number of take-home points that you can see when looking at this plot. Number one, like I just mentioned, we're an order of magnitude away in a sample of this size, at least, from observing a category that passes our multiple testing corrected threshold. We're seeing categories that show just as significant burden in controls as in cases. And some of these categories could sound like reasonable hypotheses for autism risk. Intronic SNVs in strongly transcribed or genic enhancer regions. One could easily make a story about that, except that the burden we're observing is in controls. And I think this speaks to the inherent noise in the data at this sample size. The other thing that I hope you can see is that there are a large number of categories that pass a nominal significance threshold, which I think really emphasizes the need to apply a more stringent threshold in order to observe categories of annotation that are meaningful and replicable for autism biology. Now, how we got to, how did we determine what this multiple testing threshold is? It's not the standard one from GWAS. We had to calculate it anew, because this is a new type of test. And we're estimating that among the 52,000 annotation categories possible to test in this framework, from the about 14,000 categories that we did test, because I had 7 or more variants, that there are about 4,000 effective tests. And we calculated that by looking at the correlations between annotation categories, because the vast majority of these categories are not independent from each other. So we did this by generating 20,000 simulated data sets of variants that we then annotated to the same annotation categories that I've just shown. And then we calculated correlations between the p-values from each of these simulated data sets across our 52,000 annotation categories, dropping out the categories with insufficient numbers of mutations each time, and then used eigenvalue decomposition to estimate the number of effective tests contained within the universe of tests that we had, categories that we had tested. And the plot here is showing the minimum and the 95 and 99% upper boundary on the confidence interval around the effective number of tests. And down here at the very bottom is our current sample size of 519. By combining these simulated data sets, we were able to estimate the sample sizes double R's, quadruple R's, 8 times and 16 times our sample size. And based on this calculation, we estimate the number of effective tests in a CWAS framework will asymptote around 10,000 tests. So that is determining our likely or projected multiple testing threshold for an analysis of this kind. All right, so we've tested a few obvious things: non-coding burden, burden in different categories of coding and non-coding regions near genes. We've tested categories of annotation in every combination that we could come up with, which is, we get the critique that this might be overkill. Are you testing too many things? Are you not going to be able to observe a signal? So, given that we had information from our data simulations about the independence or lack thereof between categories, we thought to use that information to identify a set of highly independent categories from within our universe of tested annotations, and then to test only that limited set of independent categories for case burden, which then would reduce our multiple testing burden. So to do this, we took our annotation categories and used the correlation information between those categories to group them into 200 clusters. And then here, this is defining the clusters of points. This is a TSNE plot of each of the categories. And they are colored by the direction of the burden that we observe in our data. So case burden is again shown in red and control in blue. We can see that categories that show case burden over and over again get clustered together, and control burden is the same. A few of these are highlighted here and further annotated, so you can zoom in and look at each of them. For example, cluster 122 has to do with link RNAs. Cluster 8 has to do with indels. 107 has to do with missense variants. And this is just to illustrate that this clustering method is pulling out coherent sets of annotation that do, at a quick glance, seem to be capturing the same aspects of biology, which we would hope they would. And then what we did is choose the category within each cluster that had the largest number of mutations in it. So each of the categories that we selected from this has the largest number of variants, and therefore would give us the best power to observe a burden And that result is now shown in this slide. So this again is a volcano plot, much like the COS plot, but here we only have 200 categories instead of 14,000. And here, again, the nominal p-value of 0.05 is shown here, and our multiple testing corrected threshold for 200 tests is shown here. This is obviously much lower than correcting for 4,000 tests, and yet we're still about an order of magnitude away from seeing a significant result in a sample of this size. But I think this demonstrates that that the sample of the size is likely too small, or our annotation is not yet sufficient at distinguishing signal from noise in a way that would allow us to observe a significant category in this analysis, no matter how many tests we are correcting for at the end of the day. All right, so we've tested single categories.
Can I interrupt for a second, because we asked a question via the chat, and I think it's connected to exactly these last slides, and I think it would make sense to have him have his voice, even though his microphone doesn't seem to work. So I will read it now. So he has the question: many categories are not significant because of multiple testing correction. That's what you meant to this point. My question is, do you think the current sample size is insufficient enough to detect the weaker signals compared to exon in non-coding region, or the negative results mean that the non-coding region is not so important? I think you actually implicitly answered this one, but I still wanted him to give the opportunity here.
Sure. So I think that the main conclusion is that our sample size is not sufficient to observe a significant burden of any one category at the current time. We have some power calculations later on in the presentation that that illustrate that, and I think that also make the point that any category of non-coding mutation, non-coding annotation that is that is associated with autism risk is not likely to be as strong as any of the categories of coding risk that we've observed so far for autism. But that will be better illustrated on a slide to come. Hopefully you'll allow me to come back to that.
Yeah, thanks. So again, if you come closer, I think then you're better to hear, but I think we got it all. Maybe I have also one short question. So just from my gut feeling, it would have meant for me, like if you, so where's the difference coming from? So first you say you have around, if you test for these 14,000 categories, you actually have 4,000 independent tests, right? That was your threshold of multiple testing that you applied here, and I think that makes total sense given the correlation between these categories and so on. But then I would have thought, like, if you then try to extract the actual independent clusters, that's what you then did for the next step, these 200 independent clusters, then I would have assumed, like just from my general gut feeling, that you actually get 4,000 independent clusters. So where's this difference between the 4,000 and the 200 coming from? Is this because you applied some certain filters for these independent clusters as well?
Right. So the selection of 200 clusters, in this case, out of a potential universe of 4,000 independent tests or effective tests.
So you basically took the 200 strongest ones.
Yeah.
Okay. That's why also this is like this direct number of 200, because that's also surprising that you have directly 200. Okay, that explains it. Okay, you basically chose these 200.
Yeah, we use k-means clustering to select this number of clusters. But yeah, I mean, you could do the same approach to get to any.
Okay, perfect. Thanks. Come closer to the microphone, please.
Okay.
Okay, somebody else here who says thanks. So he seems to be pleased with the answer.
Okay.
Somebody else? I'm also happy to read any questions. So if you don't dare to speak up, let me know and I will do it for you. Thanks, you can continue.
Okay. Great, so we have tested, we've tested single categories at different levels of multiple testing correction, and now we thought to move into tests that aggregate signal across categories. So if we can't observe a single category that we can definitively associate with risk, are there groups of categories that would give us hints about the types of annotation or the categories of mutations that are likely to be significant at higher sample sizes? And for that, we've done two different tests here. So I'll illustrate the first one here. We're calling this a cross-category burden test, and it's really quite simple. So if we go back to the result from our COS, basically what we ask here is, at a nominal significance threshold, how many categories pass this threshold on the case side and on the control side? How many categories have a nominally significant case burden versus control? And then when we run 10,000 case control label swapping permutations, how many times do we see similar numbers of categories passing above that line? So what is our null expectation? And the result from this test is illustrated in this distribution plot over here. So for case categories, at least the example here is coding regions, but this is just to illustrate what the result looks like. We see about 220 categories that pass nominal significance on the case side of the equation, but only a handful that show control burden at nominal significance. And the distribution shows the number of tests that pass this threshold when we shuffle the case and control labels. So the p-value comes from the comparison to this null distribution. And using this test, we can consider specific classes or specific groups of annotation categories altogether. So these are a few of those results. So if we look at all de novo mutations that are coding, that's the result I just showed. It is perhaps just a sanity check that we do see a strong shift in the number of significant tests toward the case side. When we look at any and all non-coding categories of annotation, we do not see a significant difference between the number of categories that pass that significance threshold on the case or control side. So this was disappointing for us to see. When we break this down in a little bit further, looking at other cuts of the data, here in the center, we're looking at only de novo indels. Our hypothesis being that an indel is a larger variant, may be more disruptive for regulatory elements in the non-coding genome, for example. And again, we're showing coding on the top. Again, so it shows a strong shift toward case side, and non-coding on the bottom. In this case, at least for non-coding indels, we're seeing a nominally significant shift toward a greater number of results than we would expect. Greater number of significant categories than we would expect. So this may be a hint of what's to come. Maybe non-coding indels will matter, but this p-value is only nominally significant. So again, if we correct for just the 6 tests I've shown here, this disappears. And we also looked at de novo mutations in and near autism risk genes. Again, sanity check, we see for coding mutations in autism risk genes, a strong shift toward case burden. And this actually is significant after you correct for the 6 tests here, which is fine. But for non-coding mutations in and near autism risk genes, we're not seeing this shift at all, which was quite surprising. So we thought this was interesting to look at and perhaps tempers our expectations a bit for the rest of the analysis. But again, may be an approach that will be fruitful, again, as sample sizes grow, being able to identify both single categories and groups of categories that are likely to have control or case burden. And the last approach that we tried on this data set within our analysis framework was what we call a de novo score. And this Again, it is somewhat analogous to the concept of a polygenic risk score aggregating signal across categories, except in this case, in contrast to the cross-category burden, where we went in and said, I want to test all non-coding annotation categories or only indel categories. We went in with an idea of what to test. Using this de novo score approach, we used lasso feature selection to identify sets of categories without our input that predict the greatest proportion of variability in the case status of a given variant. And in this case, in the 519 families, we cut the data up into fifths multiple times, and on 1/5 of the data ran the lasso feature selection and then tested those features on the remaining 80% of the data set. And by doing this over and over, we found that the average percent variability in case status predicted by the selected categories was about 0.31% here, and this is not significant in a sample of this size. So this, again, did not turn up a result of interest to us. Now, given that we were not finding categories or groups of categories that were significantly associated with autism risk in this set, we wanted to run a power analysis of sorts to try to determine how likely it was that we were to observe case burden at all, and then to try to predict at what sample size we would be able to observe such burden. So this plot here is showing the result of that power analysis. So every dot is a category again. The x-axis is the relative risk of that annotation category. And the y-axis is showing the number of mutations observed in that category. And the lines from the top, the green and the purple, are showing, are shading out what we would have had, or what we predict to have had at least 80% power to observe. The green line is showing that power at a nominal significance threshold of 0.05, and the purple is showing our corrected threshold. So going in, it would appear that we do not, again, don't have any categories that that fall into this purple shaded area. So again, it's not surprising that we did not observe any that pass this threshold. And in this plot, or on the same plot, I'm now showing two signposts or benchmarks of categories of mutations that have been significantly associated with autism risk in the past, as a way of adding some context to this. So essentially, if there were a category of non-coding variants that were equivalent to de novo loss of function in their effect size and the number of mutations observed, we probably would have observed it, or may have observed it, at a p-value of 0.05, but this doesn't pass our correction threshold. So even a signal as strong as de novo loss of function mutations, we're not going to be able to observe in this sample size. And a signal equivalent to de novo missense falls deep in this region below where we had power to observe that. This plot it takes this a step further. So the X and Y axes are the same in this case, and the red dots again are marking these benchmarks or signposts of categories of risk associated with ASD already. And the colored lines are marking off what we expect to have power to observe at different sample sizes. So 519 is the size we're at in this analysis. That is the purple line way at the top. In a sample of this size, we maybe would have had potentially sufficient power to observe a signal equivalent to de novo large CNVs, but possibly not. And what we predict is that we will need as many as or greater than 8,000 cases or families to observe a signal as strong as de novo missense in whole exome sequencing. And right in this ballpark of de novo missense are two very likely categories of non-coding risk, promoter variants, UTR variants. And so I think this puts this in context and emphasizes that we really are going to need thousands, tens of thousands of cases to be able to observe categories of mutations that are associated with autism risk in the non-coding space. That said, whole genome sequencing is certainly the way of the future, and there are several large efforts underway to generate whole genome sequencing data, process it using coordinated pipelines, and to make the data available to each other for sharing. And one example is the WGS-PD, or the Whole Genome Sequencing and Psychiatric Disorders Consortium, which is a set of groups who are collecting whole genome sequencing data and generating that and then making it available to each other. And so these sample sizes that we think we need sound daunting, but we may actually get there. I think all is not lost. And if I can switch gears slightly for one slide, I would say that I want to emphasize that whole genome sequencing data, even at this time when we are likely to be underpowered, is not a total loss. There are brand new things that we can learn from these data. So like I mentioned earlier, the Talkowski group took the lead on identifying structural variants from whole genome sequencing data, including, these are just two examples from the very large amount of work that they did on this, of genetic diagnoses, essentially, for these patients that were not known before. So on the left, you're seeing a de novo reciprocal translocation that impacts the gene GRIN2B. And on the right, you're seeing a small de novo deletion of just 3 exons of the gene CHD2. And so there are risk variants to be discovered from these data and returned to patients. And structural variants is one of those areas where the whole genome data definitely provides an advantage over existing methods. Potential to discover these small and complex structural rearrangements. So if I can summarize that, the main body of this talk, I have a few slides after this that give an update, but an update on our analyses since this was published, but I'll summarize this now. So what we see from our analysis is that there's no clear evidence of non-coding association in 519 families with autism spectrum disorder. And from our power calculations, we believe this is an issue of sample size and not necessarily of the total non-existence of risk in the non-coding genome. We can't conclude that yet. We do think, based on our power calculations and the burden that we observe, that signal from non-coding categories is probably weaker than de novo missense, or about in that ballpark. And our ability to find the signal going forward will be helped by improved annotation, so our ability to distinguish signal from noise in the non-coding region, much like we conveniently can in the coding region. And it will also be helped by the existence or the possibility of running high throughput functional screens like MPRA, which is an approach where we can evaluate the regulatory output of a regulatory region with and without a variant. And this gives us an easy output in the way that testing missense variants would not. So we may actually have an advantage here in figuring out non-coding, figuring out the impact of non-coding risk variants. And a key point across all of the various cuts and slices of our analysis is that multiple testing must be addressed in this analysis to account for the number of tests that you have explicitly and implicitly run. And we keep coming back to this analogy between candidate gene studies versus GWAS and exome. We strongly feel that you're at an advantage if you can test as many categories as possible in as unbiased a way as possible, as opposed to coming in with a candidate annotation or a favorite regulatory data set as a way of annotating the non-coding genome. We think that this unbiased approach is more likely to lead to sound and replicable associations in the future. And as we've seen very briefly on the slide about structural variants, whole genome sequencing can already today improve our diagnosis for patients within at least coding regions, particularly at the level of structural variants. Are there any questions at this point before I give a little update about our continued analyses? No?
Hui is again typing. So, while doing the tests like COS and cross categories, have you applied some filters to the variants to further enrich the potential signals? For example, use the variants in active enhancer regions only. If not, would it help in your opinion?
I think that we have, right, so we have inherently tested that. We have annotations for active enhancers, or at least markers associated with active enhancers in our COS, but I don't recall us running the cross-category burden test on specifically that cut of the categories, which we could do, but I just don't know if that matters yet.
It's not what it is.
Yeah, and that's not necessarily where the data points us to. So that's not, for example, among the categories that rise to the top of our COS analysis. I'd be more likely to try to choose a category that came to the top of that volcano plot on the right-hand side, and then you check those similar categories for a cross-category burden. But yeah, right now the data is not pointing to those active enhancer annotations.
Okay, thank you. But there's another question now coming up. I think your summary needs to be put just on slides that we share, if this is okay for you, but I think as long as there are questions, we should go to them. So Howard Edinburgh asks, your emphasis on multiple testing correction is very important, and I totally second that. But given the number of categories you look at is much larger than the number of genes, would a gene-based test work just as well? We'll still need large numbers.
Yeah, so the question, right, we will still need large numbers. And the question, and this is a key question that comes up, I think, from GWAS and from these analyses, is how do you best assign a variant to a gene, number one. And actually, you know, so we've done the number of effective tests that we've estimated from our 50,000 annotation categories is only 4,000, and a gene-based test would be about 20,000 genes that we would have to correct for in that case. So I guess I'm not convinced that a gene-based test would help us out in the multiple testing correction space at this time. But,
and again, so I second totally Howard's statement that the multiple testing correction, that you emphasize that and that you address this very fairly in the manuscript is really a very important point to get trust into these analyses in there. That's a very, very important step for the field. Maybe, so you have another 3 minutes to give your summary.
Okay, great. I will skip through it quickly. So since we submitted the paper that I've talked about today, we've been able to access and analyze data from additional Simon simplex collection families. So the first cut of the analysis was based on 519, and we've now analyzed 1,900 SSE families and run all of the tests in this annotation framework on those same families. I'm going to skip. So again, perhaps as we would predict, based on our power calculations, would suggest that we need at least 8,000 families, if not more, to see a single category that is significantly associated with risk after multiple testing correction. We don't see significant results at a significant burden for all non-coding variants. We don't see a single independent category by our clustering. By gene-defined tests, with the exception of loss of function now, because we're cutting into that set of the data that already had an observed loss of function variant. And I'm going to skip through this quickly. Again, CWAS, we're still about an order of magnitude below significance, although our number of effective tests has risen because we have a greater number of categories with a sufficient number of mutations to be tested, as you would expect as your sample size goes up. And, but what is new, what we do start to see now is a set of categories that are coming up as significant by our de novo score analysis. So in this case, we're able to use the 519 families from the first analysis as our test set for lasso feature selection, and then test the selected features on the remaining 1,400 families. And now we see that the categories selected by this lasso procedure predict account for about 1.67% of the variability and case status of the variants, and that is significant. Now, if we go and look at the categories that the lasso predicted, we can split those out into the coding and non-coding categories, and we remove the loss of function categories from this analysis, again, because this is something that we already knew going in. The coding categories out of this set of features that were selected accounts for 1.08% of the variability in case status, and this is significant. But now the non-coding set of categories accounts for 0.54% of the variability, and this still remains significant at a p-value of 0.02. When we look at the category, the specific category names within these non-coding annotations that were selected, the top terms that come out of that set are FASTCons, PhyloP, and promoters. So what this tells us is that conserved promoters, considered as a group, are those categories that are showing an aggregate burden in the case direction. And so I'd just like to point out that the promoter categories alone account for 0.5% of the variability out of a possible 0.54% from all non-coding. So hopefully that makes some sense. So as we've moved into a larger sample size, we still don't see a single non-coding category that passes our multiple testing correction, which is what we would expect given our power calculations from our publication. And still, again, of course, multiple testing correction is essential for this. But multi-category analyses are beginning to demonstrate the existence of de novo non-coding association in autism, at least at the multi-category aggregate level. And this non-coding signal appears to be strongest at conserved promoters, perhaps suggesting that as sample sizes grow, the category or group of categories in which we're most likely to observe a significant burden may be in conserved promoters. So that's the update on where we are today, and I would just like to thank all of you for your attention, and thank, I pointed out the PIs on the first slide of this presentation, but the other trainees and staff and people who worked with us on this analysis, and again, my co-first authors are bolded here, and that's it. Yeah, so if there are any remaining questions, happy to answer, but understand if we need to get off the line.
Thanks so much, Donna. That's very, very great. It was a wonderful overview here. It's already 3 minutes after the hour, so I think I have to cut it short because actually I am the host and I have to run to pick up my kid. So, but if there are questions, I'm very, very sure Donna will be happy to answer them also via email, also via the listserv or similar things in there. The slides are recorded, and if you send us the slides, we will actually share them also with everybody else. But you can probably just also go for the manuscript and just read it yourself as a repetition in here. Thanks so much again for being with us, and I hope everybody agrees that this was a wonderful presentation. Have a great morning and see you all around next time.
Okay,
bye-bye.